Last lecture in the 2013 Penn State Lectures on the Frontiers of Science. This annual lecture series is organized by the Penn State Eberly College of Science as a free mini course and as a gift to our community. We are able to provide these lectures due to the financial support of the Penn State Eberly College of Science and the generosity of our speakers who volunteer their time to give these lectures as a gift to all of us. Our theme this year is your genes, how they contribute to who you are. Our speaker today is Professor Scott Selleck. He is head of the Department of Biochemistry and Molecular Biology at Penn State. His research includes efforts to discover how interactions among molecules contribute to a number of neurological and behavioral disorders in children, some of which are epilepsy and autism. He also is working to identify fundamental mechanisms of nervous system development. As part of this effort, he hunts for novel genes that affect important developmental processes inside the brain. Among the honors he has received for his research are the Basil O'Connor Young Investigator Award from the March of Dimes and the Alfred P. Sloan Foundation Research Award. The title of his lecture today is Genome Instability. Is it the crucible of neurodevelopmental disorders in children? Please join me in welcoming Dr. Scott Selleck.
Good morning. John up on the camera has a challenge today because I tend to wander around. And he's given me certain boundaries. I'm not very good at staying within boundaries. I warned him about that already. So I have to apologize a little bit. This picture, this is what I'm supposed to look like when I get up in the morning. When I was about 12. I don't look like that anymore. I look a little bit more like this. And that's my son, Ryan. So that's a little bit, that's truth in advertising here. All right. So the title that I'm giving you, it's actually not a widely accepted view. That is for certain. What it supposes or asks is whether the fundamental root cause of behavioral and neurodevelopmental disorders in children is the instability of our genomes. In other words, the ability of our genome to replicate itself accurately. Now, genomic instability plays a very big role in another disorder that's pretty important in our society. Anybody know what it is? Cancer. Yes, sir. So in cancer, there are mutations that start in somatic cells, and of course people inherit susceptibilities to certain cancers as well. And those mutations end up creating a process where the genome changes dramatically. And those dramatic changes disrupt the processes required for the appropriate control of cell division, the appropriate control of cell-cell contact, right, which means tumor growth, tumor invasion. Right, so genomic instability and its role in disease is not a new idea. It's fundamental to our understanding of cancer. And the real critical genes in cancer are those that protect the integrity of the genome. If there is damage, appropriate repair mechanisms kick in and fix it. So I've borrowed from that idea, asking the question, is it possible that something like that is happening in these children? And the reason I've come to that is because we find a gob of genetic change in these kids. Lots of it. And I want to know where it comes from, and I don't think it's just bad luck. Okay? So there you have my whole talk in 45 seconds. But that's the fundamental concept. And I will tell you, though, that this title, hence the question mark, my fellow geneticists, or human geneticists, they would probably disagree with it. But I actually come not to this problem as a human geneticist. I come as a developmental biologist, and that's a bit different approach. And I'm going to hope to bring the rest of the community with me, but of course we have to get data to do that, and that's what we're in the process of doing. And I'm going to share with you some of that data as to why I think this is a possibility. I'll tell you why I'm so interested in this possibility. What if our environment influences the stability of our genome? For our children, in utero, our germlines. If that is true, then we need to know about that. Because if we are going to prevent these disorders from happening, we're going to have to protect the integrity of our genomes. And so I really want to know if there are environmental contributors out there that influence the stability of our genome and hence are the grist mill for these kinds of changes, okay? Because that's something we can do on the side of prevention. Amen for prevention. Can I have an amen? Amen for prevention. Because treatment is tough, right? Getting the exact right medicine that only affects that process that's disrupted and nothing else, that's tough, all right? Okay, so I'm a big believer in prevention. All right, so this is really what we're asking. Those of us who are geneticists and genomicists, are we going to, I guess I need my pointer now. Are you with me, John? He says yes. Are we going to be able to take this information about our genome and really do something useful with it, make predictions, be better at prognostication, and then ultimately to intervene? Can we do something smart? And I think it's happening for cancer treatment. We're in our early days, but we now do have drugs that specifically clobber the problem in that type of tumor. Chronic myelogenous leukemia is one of them. Gastric tumors are another, where there's one drug called Gleevec that clobbers the molecular deficit of those two disorders, and it's miraculous. These people that have this leukemia, they take Gleevec, and they might have been essentially on their deathbed, and they take this, and they're up taking Roomba lessons 6 weeks later. I kid you not. It's amazing. Now, the tumors acquire resistance to the drug, so the challenge now for those patients is develop other agents. So it is possible. It is possible to specifically target things. And that's happening for cancer, but we have a long ways to go. But I'm going to talk about behavioral disorders, and I want to start by talking a little bit about its impact. How big of a problem is it among us? Well, this comes from a World Health Organization sponsored report that says the estimate for the world, that is, right? For the estimate for the world, 2.5 trillion are the estimated costs for 2010, which, you know, I kind of look at that and I go, what does that mean? It's a big number. So I need another number to relate it to. And here's the other number. The total spending for healthcare in 2009 was 5.1 trillion. All right. Okay, the ratio there, I can get that. Half of our dollar of healthcare is related to mental illness or behavioral disorders. In the United States, $317 billion a year is what the estimated cost of these disorders from, loss of productivity. They're not out there being good taxpayers anymore. And of course, there's the direct cost of treatment, and there's disability that people get when they have these disorders. Okay, well, that's a big number too. Well, how big is it compared to other things? All right, 208 cancer costs were $202 billion. This is a big number and a big problem. And it's gotten relatively short shrift in our society for complicated reasons. Right? There's still people that believe that psychiatric illness is a weakness of character. I guarantee you it is not. When I was a medical student, I spent 12 weeks, I did my psychiatry rotation in a lockdown facility in St. Louis, ironically called Malcolm Bliss Hospital. And I saw some amazing disease, and it had a very profound effect on me because it taught me these disorders, they are devastating to the person, to the family. You know, a 20-year-old college student comes in to the psychiatric ER with the first episode of delusions and schizophrenia. And I had to be part of the group that would tell the parents what this meant. Here's your 20-year-old daughter. She has schizophrenia. And they would ask me questions like what? Do you think she'll finish school? Do you think she'll ever get married and have children? Are we going to have to take care of her the rest of our lives? Right? It is a devastating disorder. And I saw it in various forms, everything from people that were trying to do me great harm when I asked them questions for interviews to, anyway, it was a very profound experience. And it showed me how serious these disorders are and the impact they have. And they are medical disorders, no less so than diabetes or anything else. All right. The focus for my research and what I'm going to primarily discuss today is autism spectrum disorder. And I just wanted to give you a little background on that. First of all, it's a clinical diagnosis. There's no lab test we can do, no blood work that says, oh yeah, there it is, autism. It's a clinical diagnosis. And it's based on the following issues that they have. They have what I would call aberrant social interaction, and it's quite variable. Everything from a child who will not speak, cannot stand anyone touching them, to someone who's just a bit of an odd duck, right? A very wide range. They have delayed and compromised language development and repetitive and stereotyped behaviors. But they're highly variable in their expression, and they don't necessarily have all three and variations in all of them. And if you think about it for a second, these, especially the first two, aberrant social interaction and language, these are the two hallmarks of being a human being, and probably the most complex things we do. We interact with one another. We know if you come a little too close, you're in my space, man. Right? We have a sensibility of that. When is appropriate space? That's a complicated sensibility. And of course, language is another inherently human trait. So these are the things that are getting compromised. Now, what is the current prevalence of this disorder? Well, the CDC just released its data from 2008 acquired information. And this is from surveillance at 14 different sites across the country. And it's record surveillance, medical records, school records, and they peruse these records looking for the cardinal signs of autism based on the DSM-4 diagnostic criteria. And when they do that, they came up with a number this last round of 1 per 88 children. All right, 1 in 88. That's a big number. By way of comparison, childhood cancer, that is from 0 to 18, it's 1 in 800. Okay? So, this number, you can imagine the impact it has on our medical care, our schools, and the dynamic of the families, right? This is a big number, so we better figure it out, is the way I look at it. Now, the level varies with state. You know, there's some states where it's, half this. And it seems, for example, the rates in Alabama are the lowest. As it turns out, Alabama is also one of the states that have the lowest benefits that are provided for these families. New Jersey, it's one of the highest. And New Jersey has great benefits for families that have a child with autism. So clearly, it's unlikely that geography and the people in those places account for that. So there's confounding factors here. But there's one factor that is not confounding, and that is that the method of this team doing the analysis has stayed the same over time, and they're monitoring it over time. And the numbers that they did back from 2002 was it was 1 in 156. That's a good number, that comparison, because they're using the same methods. However weaknesses of their surveillance may be, those are comparable. So the prevalence is going up for whatever reason. In South Korea, they did an interesting study of a town where they examined about 20,000 people. They did a little bit more in-depth surveillance, and they came up with an incidence of 1 in 38. Now, I think the severity of what they would call a child with autism spectrum disorder is probably less. But by their criteria, and what we use generally, by those clinicians, it's 1 in 38. Now, whether or not is it higher in South Korea than it is in South Dakota, we don't really know yet, because the methods of surveillance weren't done in the same manner. But we need to do that. What are the rates in indigenous peoples? What are the rates in the Kalahari Desert among the Maasai? I don't know. I'd like to know. In fact, I'm thinking about doing that. Turns out we have a relationship with the Nelson Mandela Institute for Science and Technology in Tanzania. Guess who lives in Tanzania? The Maasai. And there are Maasai who are living traditional lifestyles, and there are Maasai who've migrated to the urban environments of Arusha. Arusha is a town of 500,000 people. I mean, I never even heard of Arusha. And they said, well, the big town close to Mount Kilimanjaro is Arusha. I go, oh, how big a town is that? I thought they'd say, 15,000. It's 500,000 people in Arusha, and the diesel pollution there is off the scale. So you can see where I'm going. Okay, is the incidence changing? $1 million question, especially if one is considering, does environment have an impact on this disorder, right? And this is hotly debated among the experts. If you ask the epidemiologists who study it in Minnesota, they will say no. The fundamental incidence is not changing. It's our ability to detect it. It's our knowledge base of it, the awareness among our community, the awareness among our educators and family docs, et cetera. And there are other people, like in California, the epidemiologists there at the Mind Institute at UC Davis, who have looked and tried to control for those factors over 15 years of study. And they say we can't account for all the change in incident from these changes in diagnosis, et cetera. Okay? So even within the community, there's a difference of opinion. My best guess is it's changing. It's going up in real terms. That's my belief, but again, there's no consensus in the community. I happen to be closely affiliated with these collaborators in California, and I know them, and they're really good epidemiologists, so I trust them, and they say, Scott, it's going up. So I believe them. But even if it isn't, it's still a big number, right? So going up or not, it's a problem. Okay, well, I'm a geneticist, and so I have to very quick succession get to this. So this is a genetically tractable disease. Why? Because genes play a big role. How do we know that? Well, several different studies have looked at the concordance rate among identical twins, monozygotic twins. That is, if one twin has it, how often does the other identical twin have it? And the answer is between 60 and 80%, depending on what criteria you use for is this autism or not. But it's high, okay? But it is not unique. You know, it's not as if this disorder is some very readily identifiable entity that is just unique to itself. You find these symptoms of language problems and social difficulties among many other disorders. Fragile X, tuberous sclerosis, which is a benign cancer syndrome. So it's not unique entity. It overlaps with a lot of other stuff. And I think you'll see why as I go on. But one thing that is true that has come out in the last 5 years, of these kids, if you do a fairly crude assay of their genome, looking for deletions or duplications across the whole genome, somewhere between 5 and 10% of those kids, you're going to find something in there that you don't find in controls, that is, in normal developing kids. 5 to 10% of the time. In other words, a big chunk of DNA missing, or a big chunk duplicated, all right? And what do I mean by big chunk? I mean something that covers many genes, 30, 50, 100, okay? And that's known as a copy number variant. And copy number is very significant. In other words, these kids, they don't have both copies of that interval deleted. They only have one. So they have one good copy, one bad copy. So it's a gene dosage thing, right? It's how much of the product of those genes is being produced. And losing it by 50% is enough to send them over the threshold, okay? So these are dosage-related phenotypes. They behave as a dominant, right? Lose one copy, one good copy, they get it, all right? Okay. So the current estimates from the research in the last 5 years, and the pace is picking up, is that there are hundreds to thousands of genes that contribute to autism. Now, to some people in the human genetics world, this was a big surprise. They're going, wow, darn, we thought it would be 20, 50 tops. It's thousands. Now, this did not surprise me. Why? Because I'm a developmental geneticist, and I work in fruit flies, among other critters. You know how many genes it takes to put together the retina of a fruit fly, right? That beautifully geometrical structure that you see when you look at the housefly? A couple thousand minimum. A quarter of the genome is required to put together that nervous system structure accurately. And I go, wow, if it takes 2,000 genes to put together a fruit fly retina, how many genes is it going to take to make sure that you can interact with everybody and know what social space you should give them? Or language development, right? I mean, behavior is a far more complicated thing in a fruit fly eyeball. And just by way of comparison, people go, fruit flies, haha. Why are you studying this stuff in a fruit fly? 75% of the disease genes that are identified, they have a relative that's functional in a fruit fly. 75%. We have about 23,000 genes. A fruit fly has 13,000. So a fruit fly is it learns. A fruit fly has the capacity to do associative learning. If you pair odors, and one odor, they're sitting on a little electric grid, and one odor, they're not, and then you give them a choice of those two odors, guess where they go? They go zip over to that odor that was not paired with the shock. And they do that 98% reliability. In fact, the first mutants identified that affect learning and memory and retention, guess where they were discovered? In wacko people like me studying fruit flies. When those same genes were assessed in mice, in spatial learning in mazes and all that kind of stuff, guess what? Same genes, all right? So the good news is that there are critters like fruit flies where fundamental processes of how you put the nervous system together, you can study them there, and they're cheap. And they reproduce quickly. And they reproduce the way you tell them to. Okay? That's difficult with human beings. All right? Okay, so I work on two organisms. I work in fruit flies and I work in humans. For fruit flies, the cage costs are very low, and for human beings, I don't have to worry about it. I love mouse genetics, but for mouse genetics, you have to provide cages and veterinarians, and they're pricey, those mice. Okay. For the sake of your understanding why diagnosis, for example, if we had a DNA test that was accurate to diagnose, why would that be useful? Well, because we know that early intervention in these children matters, okay? So this is a recent paper that just came out that showed that electroencephalograph deficits found in children with autism can be corrected by intervention. So what do I mean by EEG? So this is you take a child and you put a grid of electrodes on their scalp so you can measure, quote-unquote, brain waves. And there are certain tasks which they have a very different response to than a normally developing child. And one of them is recognition of pictures. What do you think is the most important picture that you could put in front of a 6-month-old baby that you're going to get a response to? Mom, you betcha. That's exactly right. You can also do eye tracking in these children. So they're sitting there, they're looking at this picture on a screen of mom, they go, yay, mom! And their eyes are tracking it. Those are qualities that you can measure in the laboratory, and those are not normal for a 6-month-old child who will develop full spectrum autism. Okay? Those changes can be reversed by intervention. So even something like that, something very measurable, everybody agrees this is what it is, you can reverse those with intervention. all right? Now, we've known for a while that intervention of other symptoms, you know, the measures that we have of their interaction with their peers, their language, et cetera, those too can be affected. And it's not just the intense interventions that you can only get in a few cities, where it's 20, 30 hours a week of a one-on-one interaction. But there are also community-based interventions that are the kinds of things that we would be able to have access to that have an effect. So early diagnosis matters. Right now, the average age of diagnosis for these children is around 5. If we could move it to 2, that would be a good thing. Okay. Now, there's also evidence that environment does play a role. And one of the pieces of evidence that I found most intriguing came from one of my colleagues at UC Davis, and they did a study of perinatal vitamin during and its relationship to autism outcome. So what they did was they determined the amount of vitamins that mothers were taking around the early period of development, both before they're pregnant and in the first trimester. And what they found was that there was an inverse correlation. In other words, if they took perinatal vitamins, the incidence of autism was lower. And it actually correlated with how many vitamins they took. And they thought the active ingredient that was probably being the most important in this was folate, for reasons of other genetic tests that they did in their study. It's also rather interesting, the proximity to freeways increases incidence. So now you can see why I want to go to Arusha and measure what's going on close to in these communities where the diesel fuel just make you cough your head off. So if you're within 300 meters of a freeway, when you were pregnant, your chance of having an autistic child doubles. Now, this is a correlation, right? I mean, there's a correlation between Australia and kangaroos, too. It doesn't mean, you know, that Australia blew these things out of the ground, right? So, I mean, there's a correlation. It's important to recognize the difference, but it's intriguing. Now, very recently, a study just came out from Norway that furthered this idea about vitamins, and they showed definitively that it was indeed folate supplementation, and it reduced the severity of autism incidence by 40%. And that paper is cited down here at the bottom, so when you get these notes, if you want to go take a look at it. The other thing I should say is that the people that, you know, like myself who've worked in developmental genetics, we see this all the time. We see where a phenotype caused by a known single gene deficit is influenced by crowding in the cultures. Like how many fruit flies are in that bottle? What is the kind of food that you put in there? Is it really yummy, yummy, rich food, or is it kind of, you know, not so good? So I had a postdoctoral fellow that worked with me for several years. He had characterized a bunch of mutants. He was from Japan, Hiroshi Nakato, and he took our mutants and he traveled to Japan with them, and he put them on a different food in Japan. And they had yummy food in Japan. And the lethality, this gene was one that caused partial lethality. They survived. Their lethality was put in half by just putting them on a different food. And when they came out of the pupil cases and showed up, they had a higher incidence of morphological defects in the retina and in the nervous system because they had survived. In other words, on the crappy food conditions that we were giving them in Boston, they didn't survive. Under the yummy, yummy food in Japan, they made it, and their phenotypes were more severe when they made it. Okay, so we see this in the laboratory all the time. So for us, environment playing a role in behavior, duh. Of course. But believe it or not, this is a big debate. Okay, to summarize my intro here, this is a big problem. We should try and intervene and diagnose it as early as we can. There's good evidence for both a big genetic contribution as well as environmental contributors. And so really the question is, what are we going to do? I also told you this disorder, there's probably hundreds to thousands of genes. So if you're a pharmaceutical company and you want to design drugs that tackle any of those targets, and you have your challenge is to do 500 targets, that's not so trivial a problem. 500 targets to get through FDA approval, drug testing. It's almost impossible. In other words, the more complex the disorder, medical intervention with a pill, with a drug, is very, very difficult. Now, people still hope that these 2,000 genes that are involved in autism, maybe it'll boil down to 10 pathways or something, right? Maybe it'll sift down through a fulcrum. We don't know that yet. That's a hope, okay? So, I'm going to tell you a couple stories about how I got into this business and some of the stuff we've done, and then I'll let you ask some questions. This is where we began. So I started working on this when I was a professor at the University of Minnesota, and I was in a department of pediatrics. And so I was so I'm an MD, PhD person. I have a PhD in biochemistry, but I have an MD, but I don't see patients. So I'm one of these weirdos. I know enough medicine to be dangerous. I can talk the talk with my colleagues in medicine. And my role in that department was to help promote young pediatricians who wanted a research part of their life, et cetera. So anyway, I had 2 pediatric neurologists come into my office one day and say, we found this family. And several of these children have autism. We know there's a deletion on chromosome 10, and that's about all we know. But why don't you quit pulling the wings off of fruit flies and do something useful with your life and study this? And I couldn't resist, and I said, okay, I'm going to do that. So we started studying this family, and it was all the individuals with the filled symbols have the disorder. So I think we had 13 or so affected individuals. And there was a single G-band deletion on chromosome 10. So what's a G-band? It stands for GIMSA. It's the stain. It's the stain that you put on the chromosomes to see these bands. And this is the way people have looked at chromosomes since the 1860s. And GIMSA, where did it come from? It's a dye used in clothing fabrication, right? So all the dyes that we use to stain tissues, they came from the burst in manufacturing of clothing in the 19th century. And we still don't know why this is. This pattern is the same for every person, same banding pattern. We have no clue the origins, the molecular origins of this band. And if you look at a baboon chromosome of the same thing, it's similar in its banding pattern, right? We have no clue what it is. Anyway, they were missing one band on chromosome 10. And so we started looking at it in more detail. We started looking exactly what DNA is missing on this chromosome 10 in those affected individuals. And they were missing about 7.3 million base pairs. And there's about 30 genes in there. So they were one good chromosome, one deleted chromosome. But what was really interesting to us was what was happening at the boundaries. So when we took DNA probes around the boundaries and we then hybridized those to the 2 chromosomes from that patient, we got weird patterns. We got this. So here's the experiment. You take a DNA probe, something you've cloned, and you label it with a fluorescent molecule, and then you hybridize it to the chromosome, and of course, because of the hydrogen bond pairing between complementary sequences, it will only stick to the identical sequences on the chromosome, okay? And so we were looking for those sequences that were deleted. Of course, it wouldn't show up at all on the affected chromosome, and you'd get a band on the normal chromosome. Here, we were using probes from the junction point, and we would get this for the normal chromosome. So we'd get signal in 2 places, which was presumably the endpoints, and then we'd also get signal from somewhere else near the centromere. And in the deleted chromosome, these 2 things had merged. So what did this tell us? It told us that there were related sequences found here and here, at the other end beneath this picture, and also up here. In other words, there were sequence repeats that were at the boundaries. Okay? Sequences so similar to one another that they would recognize one. Okay? And so we characterized the deletions, and these are the repeat sequences that were found at the ends. All right? So this block of sequence is damn near identical to that block of sequence. The colors show you the related elements. That purple block related to all those purple blocks. So there are 95% identical sequences at the junction points. So what does this say? It says, well, it's likely that these rearrangements are affected by the sequence organizations at the ends. And somehow this is promoting rearrangement. Okay. And it turns out it does, and it does at many locations. One of them is on chromosome 10 that I described for this family, and we've now identified about 50 other families that have it. But this is now, these kinds of changes where you get rearrangements around these sequence repeats, they've been called genomic disorders. They are recurrent disease-causing sequence rearrangements, that is deletions or duplications, inversions, etc. And these rearrangements are associated with sequence architectural features that are known as segmental duplications or low copy repeats. Those things that were at the ends, okay? Now, what are they? They're big sequences. They're greater than 10,000 base pairs generally. They are found oftentimes, between 50,000 base pairs and 5 million base pairs from each other. And basically what happens is rearrangements between these two things take place because they're so similar. Okay, so these are the characteristics of segmental duplications. One other important feature of them is that there are a modest number of them throughout the genome. There are other sequence elements in our genome that there are zillions of copies. Little teeny things, lines and signs and stuff like that. There are tons of them. These are not those. These are big sequences, moderately repeated. Now, if you look at all such sequences in the genome, what's their distribution? This is their distribution, where related sequences are connected with these blue lines, okay? So like here's chromosome 16. Look at all the related sequences there. Chromosome 22, this region right here, all kinds of related sequences. And there are about 169 regions of the genome that have this characteristic. And essentially all of them are going to be hotspots for churning of the genome and consequences of that churning, if you will. At the time of this in 2002, we already knew that there was more than 30. Now these sequences, they're not, so what I'm telling you is these regions of the genome that have this kind of sequence architecture, they are prone to rearrangement. And that can be disease causing. They also have an impact on evolution. So if you look at their distribution in different critters, like if you look at the distribution of these things in a mouse, it's really simple. There are fewer of them, and they're all found right in tandem repeat. In other words, they're not spread out. Because if they're spread out and stuff happens between them, you get mutation, right? So the mouse is really simple. Rabbit, shark. The segmental duplication pattern is really primitive. Well, simple. Maybe not primitive, simple. In human beings, it's a mess. And some people have therefore theorized, well, let's look at the structure of these sequences through evolutionary time among the New World monkeys, the great apes, etc. Is there a relationship? And the answer is there is a relationship. So here, for example, is chromosome 16 from humans, and the distribution of different segmental duplications throughout chromosome 16. And here is baboon. They're there. The same sequence elements are found in the baboon chromosome, but there are a lot fewer of them. And so if you look at this and you say, well, when were the times in evolutionary history where there were big divergences among our lineage? Guess what? Those were times when there were explosions of segmental duplications appearing in the genome. Okay? So the idea being that these sequence things, they make our genome unstable, but they also produce variability. And variability is what you evolve with. I always find it amazing that the human species, it's only 200,000 years old. We, as an identifiable species, are only 200,000 years old. Now, we also have a huge reproductive loss. How many fertilized eggs actually implant and make it to kid? Less than half. Compare that to a mouse or a rabbit. We are hopeless. We're hopeless. But we invented the gun, so the rabbit has a disadvantage. Okay? So the good news is The variability has allowed us to have, I don't know, opposing thumbs, big motor cortex, the language area, et cetera, and a sense of humor. The bad news is there's a consequence of this instability. One of them is reproductive loss, but we've seemed to have overcome that by our habits, since we're overpopulating the place at a significant rate. Okay, so how do these sequences do this? And I'm just going to give you sort of the cartoon molecular biology version, which is the way I think. So imagine two chromosomes here, and there's a gene in the middle of two of these related sequences. Well, what if the homologous recombination machinery makes a boo-boo? Right? Homologous recombination is where you actually exchange chromosome arms at sequences that match precisely. Right? So it's a method of producing variability in our genome. Homologous recombination that takes place to produce our gametes. Well, what if that makes a mistake? Instead of this tan one matching up with tan one, it matches up with this one. The product of that recombination is a duplication and a deletion. Okay? So basically, the similarity of these sequences to one another promotes mistakes in the normal machinery that does stuff to our genome, like homologous recombination. Okay? That's one mechanism that could be having an effect here. Okay. Now I want to tell you a little bit about what is the kind of genetic change that we find among us. Why are we different? And we've gone through a real revolution in this idea in the last 7 to 8 years. We used to think that the difference between us was all what we call single nucleotide polymorphisms. Single nucleotide polymorphism, in the vernacular of Clinton, who called the Genome Project the Book of Life, right? We sequence the human genome, and we can now put it on a table. And it's a sequence of A, T, Gs, and Cs, right? So in that vernacular, a single nucleotide polymorphism is a change in one letter. So you might have an A, and I have a T there. As it turns out, we have great ways of measuring those, but they're not the most significant difference between us. As it turns out, it's this thing that I've been telling you about, these deletions and duplications. They are much, much more common. We just didn't have great methods of measuring them. And of course, there are other copy number changes that are really drastic, like you duplicate the whole chromosome, right? So this is sort of a gradation of frequency of events and how big it is, right? So this is a very important type of variation that has received relatively modest attention compared to those other ones. So in the Book of Life vernacular, a copy number variant is you're missing a whole paragraph, or you've transposed that paragraph from this chapter way down to that chapter, okay? So there are different methods for assaying these changes, and I'm just going to tell you about this one, which is array CGH. It's comparative genomic hybridization, and this is what we use to measure this. How is that done? Oh, before I go there, though, here's the main point, right? Between any two humans, there are more base pair differences due to these things than SNPs, and 95% of the NIH dollar has gone to measuring this, just so you know. I'm trying to change that. I'd like more of the NIH dollar. All right, so how do you assay these things? Well, what you do is, since we've sequenced the human genome, we can literally put little pieces of it down on a glass slide. And right now, on one glass slide that you can hold in the palm of your hand, literally, we can put down 4 million different little spots, where each one of them has a different sequence. 4 million. It's a lot. So, and these are single-stranded DNA molecules. They're usually 50 to 70 some nucleotides long, okay? And they're single-stranded, so they're available for complementarity for hybridizing other DNA or RNA molecules, okay? So, we've got all these little sequences that represent little bits of the entire human genome out on this glass slide. And then we take our samples, and one sample is our reference, quote-unquote normal human DNA sample, and we label that DNA with a red fluorescent molecule. And then we take our subject of interest, and we label them with a different fluorescent molecule. And we take both of those samples, and we put them on that glass slide, and we let the DNAs hybridize to the target here, okay? Now, if the amount of DNA of any region in this sample and that sample is the same, the color intensity will be the same. So you'll get, green, red, you get yellow. So you can measure the intensities that are coming off of each of those 4.1 million little dots with your groovy camera. So if the sequence intensity is the same, then the copy number between those two samples is the same. If one of them is stronger than the other, that sequence went bye-bye. Okay? So it's a balance. So that's why it's comparative genomic hybridization. So that's how we do it. And this is the kind of graph that you get out of these machines. They'll say, okay, here's chromosome 16p11, and this looks good, nothing there. And then all of a sudden there are a bunch of probes that represent this sequence, and they show a loss of signal from your sample. Here are some that show gain of signal for your sample. So this is what deletions look like. And these actually are representing on the y-axis here the ratio of the fluorescence intensities. And there's a probe, little DNA sequence represented on the array here for each one of those little vertical bars, okay? So that's how it's done. So, summarize this. The human genome consists of a lot of this kind of difference, deletions and duplications, and they are not randomly distributed throughout the genome as I showed you in that map of where they all are. And the sequence architectural features influence the rates of them occurring. Okay. So, what is the origin and impact of these different types of genetic variation I've described? Well, this is this is the way I think, very simplistic in cartoons. If you have big things that take out lots of genes, a single event of those is likely to be sufficient to cause your phenotype, autism, developmental delay, epilepsy. And penetrant means if you have that genetic change, how often do you get the phenotype? It's going to be highly penetrant. You get the genetic change, you're going to get the disease, okay? Smaller events, everything goes down. You may need multiple events to get the same outcome. On down the line. And all of these things that influence copy number, they will be influenced by genomic stability. Our ability to replicate stuff, to not have this mispairing and aberrant homologous recombination. All those things will be influenced by the ability of our genome to replicate accurately. Now, SNPs are very small events, and their effects are going to be proportionally small, and they are. And that's where most of the genetics research has been. It's been on SNPs. Okay. So I'm going to summarize here some data from schizophrenia. Well, actually, before that, I'm going to talk about the genetics of neurobehavior disorders in a bit broader scope and say, well, how are we doing? How are we using the tools to measure this, and what are we learning from that? All right. So this graph, it comes from a great review. If you're so inclined, I highly recommend this review that came out just last year. On the y-axis here, we have heritability. That means if you get the genetic change, are you going to get the disease? And on this axis is the prevalence. And here's autism, there's schizophrenia, there's bipolar, there's compulsive disorder, there's attention deficit disorder, type 2 diabetes by way of comparison, BRCA, which is a cancer susceptibility. Okay, so you can see that there's pretty high heritability for these guys, right? Which means, as geneticists, we should be able to go to town on them, okay? So they have substantial level of heritability. We should be able to go after them. How have we done? Okay. And I'm going to show you the data from schizophrenia. And I will tell you, it's kind of disappointing. It's disappointing to me. The guys who were presenting this at a conference were high-fiving each other all over the place, but I was kind of going, and I'll show you why in a second. Okay, so here's another graph. This is the effect size. If you have a genetic change, what's the impact? What's the likelihood that you're going to have the disease? And this is the frequency, okay? So these guys over here are the ones that have big impact. They turn out to be kind of rare. right? These are the rare guys. All these blue diamonds, guess what? They're all copy number variants associated with schizophrenia, all of them. They're rare, which means if you find them, you know that the likelihood of that person getting schizophrenia is high, but you don't find them very often, right? Okay, these little red dots down here are the ones that are coming from the SNP analysis, looking for single nucleotide polymorphisms, where we have wonderful tools to do it, okay? But look at the effect size. It's minuscule. These are all clinically irrelevant. Okay, and the meeting that I was at in Germany in the fall, I think there's 12 here. It's now 61. There's something like 60,000 patients were done to get 61 genes that we now know can contribute to schizophrenia. They're all clinically irrelevant. Okay? So, in the middle is a big hole. And there's a big hole because we have technologies to do this, and we have technologies to do this, but we haven't applied any technologies to do the middle. Okay, so all these bazillions of dollars we've spent on schizophrenia research and 60,000 patients, how much of the heritability can we account for? 6%. As a disease, it's 80% heritable. And with all that stuff, analyzing 50,000 cases, that's a lot of DNA samples through your lab, let me tell you. 6%. That bums me out. And it also tells us the size of the target for schizophrenia is going to be thousands of genes. And we're back to that problem. How do you target with a medicine thousands of genes? Okay. Okay, I don't want that. All of a sudden, it's doing something strange. Okay, now we're back. So we're interested what's in the hole, what type of variants, and why have they escaped detection, and let's see if we can capture them, and that's part of what we were interested in doing. So I'm going to give you about, oh my gosh, 2 seconds worth of my own data. Anyway, how hungry are you? You're going to run out here for lunch at about 12? My guide. Okay. You still got some juice left, guys? You still with me? Okay, I don't see anybody meandering out the back door. When I was teaching medical students, when I saw people go out the back door, I knew I better shut it off. Okay, so we looked at this picture and we asked a couple very simple questions. One is there a difference in the total amount of copy number between kids with autism and not? If genomic instability is really part of this gig, we should see more change overall in these kids compared to controls. Not just looking for the rare events where you go, oh wow, bad luck, but is there more total genetic change of this type in these kids? And if so, where is it and what type is it? Is it mostly deletions, duplications, translocations? What is it? And where is it? Is it located in those parts of the genome which we know already are susceptible to change? And is there a relationship between how much change and how bad the phenotype is? Because if this genomic instability model that I started with has any validity, you would expect the more junk you see, the worse the outcome, right? So we wanted to do that. And believe it or not, the community has not measured copy number in this way. They haven't actually totaled it all up, measured it, and say how many base pairs of deletion and how many base pairs of duplication. So our study design, I'm going to go through this very quickly. We basically took samples that were generated and assessed from our colleagues at UC Davis at the Mind Institute. And so this was a really true population-based case control study. In other words, they obtained children with autism for study, and then they went out into the general population and contacted parents based on birth records and evaluated the children from the same geography and born at the same time to match the children that were with autism that they'd evaluated. That's almost never done, okay? It's expensive, time-consuming, right? You have to go out to the population, call everybody up on the phone, say, hey, could you come in? We'll give you a couple $100, and can you help us out? It's expensive science. It's the only population-based case control study in autism I know of anywhere in the world, and these guys did it. When I heard about it, I went, you guys are who I want to talk to. And they kindly provided the DNA samples for us. So we had about 500 kids or so in this study, and we looked at events we focused our attention initially on those regions of the genome that are highly unstable. That's where we put our sensors. But we also did a high-resolution analysis where we looked, so the first analysis, we could see big events. 30,000 base pairs or bigger. The second analysis, we used a tool that allowed us to look at 1,000 base pairs, so 30-fold more sensitive, just to see what we'd see. All right, so I'm going to give you the take-homes. The MIND cohort is this group from California. So the first thing I'm showing you here, on the y-axis is the percentage of the population of study, and on this axis is how big is the copy number variant. Okay, so these graphs represent different populations. This group is actually a group that has not autism, but severe intellectual disability. All right? These two are two different groups of autistic children evaluated by two different teams. They're very similar. And then these two groups are two different sets of controls. So what does this say? Phenotypic severity, this is really bad. This is not as bad, and this is normal-ish. There's more large, bad copy number events in these guys, right? More of it. Okay. I should say, a lot of this data comes from a gentleman named Santosh Girarajan, who we recruited to Penn State from the University of Washington. And he is now a collaborator, and his lab is right next to mine now. It's one of the joys of being a department head is you can recruit great people, and then you can say, hey, would you work right next to me so I can learn from you? And he said yes. God bless his cotton socks. So we then looked at what is the nature of this variation. And we just split it up into duplications versus deletions, and then we got something of a surprise. We found that these kids with autism, they had more dupes, not deletions. Deletions were the same. And we looked at a different set of autistic children, evaluated by a different team, and we got exactly the same thing. Okay, so the copy number burden, that is how much of this genetic change we're measuring, it's represented as duplications in the autism population. Okay, why might that be? What do you think? Why would duplications be more common? If you delete something, is good stuff going to happen? No. When you delete genes, in general, it's not a good thing. If you duplicate something, is it a given that it's going to be horrible to... Not necessarily. Maybe you just give a little bit more of it. That may not be so bad, you know? Maybe more is better. I think it was Mick Jagger who had said, too much is never enough. Okay, it's deletions that are not found in this group. Now, we also, so when we sent this to review, our colleagues said, you guys are out of your mind. This can't be true. So they asked us, of course, for more data, more data. When you propose something which is out of the norm, the natural reaction of the community is, show me more proof. So we looked at some data from, not our study, but from another study that was published, and this was a group of kids where it was not a case control study where you've sampled from the normal population and then you sampled from the kids with autism, but you've actually taken families where you have mom and dad who don't have the disorder and the child does. So you have unaffected parents, affected kid, and sometimes you might have an unaffected sibling. So what we did there, we took their data and we looked at what is the ratio of total base pairs of duplications in the affected child compared to the mom or the dad. And the bottom line is there's 7-fold more duplications in the kid. 7-fold. Deletions, it's 2-fold. Okay. So that's a big difference. So the kid has 7-fold more stuff going on than the mom or the dad. And this ratio, again, reflects this preponderance of duplications. Okay. What about this correlation with clinical measures? And we've been able to do that too with a test called the Vineland score. The correlation's not great. It's minus 0.2 is the best, but it's certainly highly significant. So what we're doing is saying, if you do badly on this clinical measure of how bad your phenotypes are, do you have more copy number burden? And the answer is yes. Okay? Other tests did not show it. And as it turns out, this test, the Vineland test, it's actually an interview of the caregiver. So a psychologist will sit down with the parent and interview them about, well, what was Johnny doing when they were 2? What were they doing when they were 1.5? How did things go in kindergarten? You know, so it's a broad, it's attempt to get an idea of how this child is done developmentally. The other tests that people usually run, they're done in a 45-minute window or a half-an-hour window on that kid once. I would argue that you're probably going to get more sensitive information if you talk to the parents and say, what's happened in the course of this child's life thus far? And that was the measure that gave us a correlation. The other thing I will tell you is most of this copy number change, as you would expect from what I've told you, it's surrounding those regions where there are those segmental duplications. So that's the crucible. This is the variant part of the genome that's churning away, and it's generating some of these diseases. I'm going to go through this very quickly. We did this analysis where we could see things 30-fold smaller, and we got exactly the same thing. There's more duplication burden. And this is an important point, so I'm going to talk about this a little bit. This looks at not only autism. The children with autism are the blue bars. The controls are in black. This is the amount of duplication or deletion or total, where we've added them both together, for these 3 conditions: normal kid, child with autism, or child with either severe intellectual disability or multiple congenital anomalies. So let's just take a look at this graph because it's easiest to see. There's an increase in how much copy number burden is occurring. Here's normal kids, autism, kids with severe disability. If you look at it for duplications, there's even more duplications in these kids. And there is also significant difference in deletions. In other words, the worse your neurological problem, the more copy number yucky stuff you've got, and deletions begin to show up in the more severe cases. All right, so here are the take-homes. Neurodevelopmental disorders are associated with increased variation of this type. In autism, it's predominantly duplication. The more severe the disorders, the more of this change we see. The variation is in those unstable segments of the genome, and the clinical severity correlates with the amount of it. And we're hoping that with some of these methods, we're detecting what's in that hole, some of those previously undetected variants. Okay, so now I've got 2 more slides and I'm out. I'm going to try and pull it all together. So, if you talk to the people that study this problem, there are 2 somewhat oppositional models of how it works. The geneticist says, look, it's genes. You mess up the DNA and you get the disease. And that's all there is to it. Doesn't matter what environment you can, it's genetics, man. It's all genetics. And they're right to some extent. Its heritability is 0.8, but that doesn't mean environment doesn't play a role. The other model, of course, comes from people that say, no, it's the environment. We're getting exposed to stuff. These children in utero are getting exposed to stuff, and those chemicals, disruptors of development, can affect patterning and brain patterning, and that is what causes it. And of course, the right view is it's going to be some combo, right? This plus sign, people actually argue about this. There's some people, no, it's just this, no, it's just that. But duh, it's going to be both. What I've told you about is another element of this, or what I'm hypothesizing is an important element to investigate, and that's this one. The environment can affect the stability of the genome, and that affects your genes. And the reason I care about that is because we can intervene there. If this is true, then EPA is going to have a different role in our lives. And I have an assay that can measure this type of variation. So what I want to do, I wish I could assay all of you guys and put it on a map and look at where you live and what you've been exposed to and what medical things you've had and say, is there any relationship between copy number change and your exposures and your medical history? So I want to do geogenetics. But I need big numbers. I'm probably going to start with geogenetics in Tanzania. I hope. So, what I've told you is that duplications are predominating in autism. If you have duplications and deletions, you get more severe things. And clearly, your ability to replicate your genome is going to influence all this. But what I haven't said, which is important too, is that there's going to be selection in here too, right? It's not just about degeneration of these variants, but they're going to be selected. But what I'm adding on to this is that environment is something we should think about. And we should start assaying the integrity of our genomes in the general population in different types of places. I should probably go to Beijing or Seoul, Korea, right? Serious air pollution there, and start doing assays, and that's what I want to do. In my spare time, when I'm not doing department head stuff. Anyway, I want to acknowledge the people who've contributed to this. It's many and many institutions. I have some wonderful colleagues at UC Davis, the University of Minnesota, which is where it began, and the Minnesota Medical Foundation, which actually gave us our initial funding. Two of my collaborators at the University of Washington, and one of them I was lucky enough to recruit here to be a faculty member at Penn State. Marilyn Ritchie is she spoke here. You guys know Marilyn, right? Yeah, we do. So she was the first recruit that I nabbed as department head. She's great. Santosh and my lab, and then our funding from Cure Autism Now and the Department of Defense, as well as the Institute for Environmental Health and Safety. So here's some online resources if you're interested in them, which will be part of your handouts when you get them. And here's some of the people. There's Evan, although I never see him in a white coat anymore. This is all, Sure, Evan. There's Santosh looking Santoshi, and here are my collaborators in California. Isaac is a toxicologist, molecular toxicologist. Irva Hertz-Pachotto is the epidemiologist, and Flora does everything. She runs all the DNA samples and coordinates it. And here's Marilyn and me when I was 12. And of course, it's always about the people. And these are some of the important people in my life. This beautiful gal died of cancer when she was 42 on December 19th. And this is my lovely daughter with her. So this reminds me, we have a lot to go, right? We failed. We couldn't beat back her colorectal cancer from 3 years. We tried. But we have a lot to go. There's a lot of good we can still do, and we should get out there and do it. And of course, this reminds me that all of what we do in science, it's about the people. And there's some other my favorite people, my son, and these are actually Heather's kids, Paige with her little Penn State hat on, and her son CJ at Christmas time. Anyway, with that, I will close, and thank you for your patience.
Hold up your questions real high, please, and before I start reading them, we have had some requests for perfect attendance certificates. If you have attended all 6 lectures, I have some down here, and there are some on your way out, and if we run out, just let us know. We'll make some for you. So, Dr. Selleck, to start the questions, please say a little bit more about the research studies that you would do in the next few years if you had enough money.
Wow. Well, I'd like to do the same analysis for schizophrenia to determine if there's a relationship between the degree of this type of genetic change and schizophrenia. I'd like to do it for kids that have developmental delay and not autism and see what their relationship is. And I'm really, really keen on knowing, is there an environmental contributor? So can I measure different rates of this type of variation in relationship, for example, of exposure to air pollution? So if I had unlimited resources, that's what I'd be doing.
It's funny, this next question is directly related to your answer. I have heard that there is a relationship between autism and schizophrenia, that they are opposites along a spectrum. What can you tell us about this relationship?
So what I can tell you is that a lot of the genes that are associated with autism, they're found associated with schizophrenia as well. They're certainly largely overlapping sets. There may be some that are going to be unique to each of those disorders, but they are largely overlapping. And I'm going to throw out another radical idea, and that is that these disorders, schizophrenia on one hand and autism spectrum disorder on the other, they're really not discrete disease entities. Not at all. We recognize them, we see them clinically, and they fall into patterns, so we call them something, we name them, we have to. How are we going to help these people? But as a developmental geneticist, for example, I go back to some of our developmental biology. It takes 2,000 genes to build a fly retina. And when you can very readily screen for those because you just look to see if the eye becomes rough and ill-patterned, just under a dissecting microscope. But we don't call that rough eye syndrome, right? We call it, it's a rough eye, and it takes 2,000 genes to do it. I think in the world of autism and schizophrenia, we're calling these as discrete disease entities, and they are not. They're not discrete disease entities. It takes a lot of genes in schizophrenia for us to have the normal perception of reality, the normal interaction with one another, and even more so in autism, language, social interaction. So on one hand, we have to define them in some way so we can study them, but genetically and developmentally, these are not discrete entities, and they're going to largely overlap. Developmental delay, autism, attention deficit hyperactivity disorder, the genetic basis for those, they're going to overlap big time.
What mechanism would allow duplications to be generated so much more in humans than in other species?
Very good question. So you could also ask that question, are there mechanisms that have a duplication event as a predominant outcome? And the answer is yes. I think the reason we're seeing it a lot in humans has to do with that sequence architecture. So the pattern of segmental duplications in humans is far more complex than any other species we've looked at. Now, exactly, we don't know if the duplications are showing up because they're generated or are they generated and then selected for, right? But there are molecular mechanisms where a duplication event is predominant that we know about, that we can study in critters like mice and fruit flies and stuff like that, and that's useful to do. What kinds of insults and what kinds of replication errors generate duplications more than deletions? That's useful to know as well, but we don't know very much about it.
Are you familiar with 22q13?
Yes.
Okay, deletion syndrome. If so, what can you tell us about this?
So 22q was one of those regions I showed you on that genome map. It is the San Andreas fault of the human genome. There's tons of segmental duplications in there, and deletions in that region have been associated with schizophrenia and autism, okay? So it is one of the regions that is the most variant in the human genome. 1 out of 2,000 live births have a deletion or duplication of that interval. 1 in 2,000, okay? Now, what's wild about it is that sometimes you can have a deletion of that interval and the child or the individual gets schizophrenia. And sometimes you can have a deletion of that interval that's very similar and they have something called velocardiofacial syndrome, a morphology syndrome. We have no idea why. It's the complexity of, these are genes where clearly other events are happening. But so that is one region of the genome where changes in it can contribute to these disorders, but it can contribute to others. It also shows you that the genetics of behavioral disorders, they are not totally discrete from the genetics of morphological disorders. It's all development. And guess what? The same mechanisms are used over and over again. The molecules that put together your hand, they put together your brain, okay? They're not different. It's the same mechanisms with some tweaks and twists, but the fundamental signaling systems that are used to pattern your hand are patterning your cortex, okay? So that's why there's so much overlap. Yes, ma'am.
Can modern technologies that cause less human interaction, such as television and video games, be causing autism?
I have no expertise on that whatsoever. I can say that the interventions, I talked about how to intervene on these children that have autism. How do we promote better social interaction and language? The way that's done is we engage them in social interaction and language, right? They sit that kid down in a chair, and so a child who's not responsive to anything, they start off by saying, okay, here's Starburst, and if you look at me in the eyeball and say hi, you get a Starburst. Repetition, right? So things like that, as simple as that. Or if you stay in your chair for 30 seconds, I'm going to give you a Starburst. Okay? So repetition, engagement, language development, that's what helps these kids out. It's not rocket science. It's exercising, just like, folks that have a stroke and they lose motion, right? There's a lot they can recover by what? Working on it, getting it going, right? They repattern the nervous system. The nervous system kicks in. It's very plastic, and so you start using other parts of the brain to start using that hand, etc. The same is true for any skill we have, right? We are going to be better apologizers if we practice it. We will be better at forgiving one another if we practice it a lot. Same is true for autism. Sorry, I'm waxing philosophical.
I track a fair number of children, I am a child psychiatrist, with identified duplications, deletions on array CGH that are underlined, not associated with defined disorders. Is there anyone locally studying these children?
There are clinically available tests that are fairly sophisticated for analyzing these kinds of variants in these children. The one that I know best is in Washington State. It was founded by a geneticist from Baylor, and they have a huge repository, a lot of expertise. They update their array at a very high frequency. So around here, I don't know of anyone that's doing that. In Washington State, there's a company that does this very readily. So if you have a patient that you'd like to have evaluated that way, you can easily refer them to them, and you put the DNA in a tube, send it off to Washington, and you'll get the result back in a week. And they're really good. So they provide the most updated assay of this kind of variation that there is that's out there.
Is it possible that back in the day, what we are currently calling autism was then called mental retardation, and that the prevalence has not really increased as much as we have just renamed it? For instance, what was the role of mental retardation in 1990?
So that is the argument that a lot of epidemiologists believe, that our perception and labeling of these children has changed. Our attempts to separate that, in other words, looking at current data and what percentage of children are labeled as mental retardation versus autism, we have not seen the shifts that model would predict, right? So the prediction would be we'll see more autism, less mental retardation, right? Because they're shifting out of this broad category to this other category. I think the true answer is that there is some of that accounts for the changes in incidence. If it accounts for all of the change in incidence, even the experts argue about it. So it's hard for me to give you an answer. I think it's truly on the increase because the epidemiologists I know the best who are trying to control for such things argue that it's still on the increase. This is based on California birth records, which are by far the most accurate and detailed that are available to us. But that's to that population. That refers to California. Maybe that is different than Minnesota. Maybe everybody leading that clean life in Minnesota has a good effect, where those wild guys out in California, who knows what they're doing?
Is there any evidence that endocrine disruption, for example, BPA exposure, causes genome instability?
Not that I know of. But it's a good question. I don't have a good answer to that. I don't know of any evidence that that's the case. I do know that, for example, methylation patterns. Remember I told you that folate was protective? Well, folate is a methyl donor, and DNA gets methylated. And the more DNA methylation you have, the less copy number change happens. So there's a possibility that this folate intervention is protecting the genome from copy number variation. That's purely speculative. But I don't know any evidence of these endocrine disruptors affecting copy number change directly.
Two related questions. Regarding autism detection in infants, how soon in the child's life does intervention work? And also, at what age is it too late? And then the second related question is, what would a preventative intervention look like?
What would a what intervention look
What
like?
would a preventative intervention look like?
Well, I'm not sure there's a preventative intervention. There's, so let me get to the first
Probably
part.
means remediation.
Yeah, let me get to the first part of the question. It's never too early to intervene. There's always things you can do and engage. Just like you always engage your child as soon as they're born, right? And they start smiling and you tickle them and all, so it's never too early to intervene. When do people notice it? They notice it often when they're expecting language, right? They notice it when certain motor skills may or may not be there. But oftentimes children with autism, they have perfectly fine motor skills and it's language. So that's one of the reasons that it's detected rather late. But it's always good to ask, right? Because, and again, this is a spectrum disorder. It's not like, oh boy, this kid has autism and this kid doesn't and there's nothing in between. There's all kinds of gradations, right? And so if you see that the child has some language issues or they have social, you can get on it right away, and it does help. It has an effect. So early, if there's a suspicion, consulting your pediatrician, et cetera, and intervening early is a good thing to do.
It's very disappointing always that we can't get through all these good questions. So maybe we have time for two more.
Okay.
Is there any research looking into genomic instability related to dyslexia?
Yes, there is. And this was work done by my colleague, Santosh Gavirajan, and the level of copy number variation among the children with dyslexia was normal. Okay? It was no different from controls. And I think that makes perfect sense along this gradient of deficits we're talking about. Dyslexics, it's a fairly specific and modest deficit, right? They don't have broad problems in behavior or intellect or language or cognition. They're good to go, these kids. They just have this one annoying, troubling thing that you have to help them with. But with dyslexia, in some ways that's a control. They do not have any more copy number burden by these measures that I showed you as the controls. And in fact, when we looked at them in detail, they actually were a bit lower than controls. And when we looked at why, we found that the people that were screening them took very high IQ children. So to make sure that the diagnosis of dyslexia was accurate, they'd say, we're going to take kids that have really high IQ, but they have dyslexia. Those are the people that went into the grouping. That same screening was not done for the quote-unquote normal controls. And guess what? The children with dyslexia in that study that Santosh published last year, they actually had less, barely, but less copy number change than the control groups where we hadn't done any such selection.
Why is autism greater in males?
Oh, man. Because men are hopeless. I wish I knew. My answer as a developmental biologist is that there are certain traits and qualities of being male that, for reasons we do not understand, promote or predispose to these behavioral problems. Very interestingly, a study was published last year that looked at autism in girls. And they found for having a similar level of deficit, they had almost twice as much copy number change as a guy, okay? So the girl had to have more hits to end up at the same point. Of course, this is telling us what we already know. Women are the best. Okay.
Let's let Dr. Scott Selleck know how we feel about his lecture. Thank you, Dr. Selleck.